Okay, we'll make a start. So thank you everyone for joining. So this is our Automata hosted webinar on next steps in genomic workflow automation, integrated nucleic acid extraction and QC solutions. My name is Alice. I'm a senior application scientist at Automata. My role in the company is to work with customers, look at their protocols, understand their requirements, and then translate that into a workflow using our platform. So the things that I'm going to cover today, I'm going to talk a little bit about what we actually mean by integrated automation and how we enable that. And then also I'm going to use this time to go through some of our example solutions that we've developed here at Automata, specifically at Genomics Applications. There will be time for questions at the end. So if you want to drop them in the chat box, I'll answer them at the end. I think that's the best. Yeah, we'll do chat box at the end. So in order to explain what I mean by integrated automation, I think it's a good place to start is to talk about what I don't mean by integrated automation. So what is automation without integration? So you'll probably hear a lot about me talking about benchtop automation. So what that is that's separated bits of equipment that might have automation within their capabilities, but they aren't linked. So this image kind of shows what I'm talking about. So it's 3 distinct bits of equipment. They could be a liquid handler, they could be a benchtop extractor. And, they've probably increased the efficiencies of these laboratories. So, it will increase their throughput, it will increase their hands-off time, but it still requires a certain level of machine tending by the scientists. So what I mean by that is something that's very personal to me, is you might be running something like a library prep on a liquid handler, but you are still required to tend to that machine. So that might be taking a plate off, sealing it, centrifuging it, unpeeling it, putting it back on the machine. And sometimes these are very inconsistent in when these happen. So it's not like every hour you need to do this. It might be you set up your liquid handler and then in an hour you have to come back and do some machine tending, but then it might be 15 minutes and you have to come back. And the problem that I always found with this was it kind of created this thing that I refer to as like scientist dead time. It kind of scatters your focus. So I personally wouldn't be able to go away and do something useful in 15 minutes. Maybe that's a reflection on me, but it's difficult to get your head into something else. So it kind of becomes dead time. Another thing another issue I've had with this as well, it can make it really difficult to scale. So if you're running at full capacity with your current benchtop automation, then the only real way that you can increase your throughput is by getting more of the same thing. And that often requires a lot more space, and more often than not, it will require more scientists to then tend to these machines. So you can scale, but it's not always practical within your lab, your budget, or just like the staffing that you have. So this leads me to talk about what actually is integrated automation. So this is kind of an abstract way of showing it for now, but I will get into way more detail. So when we talk about integrated automation, we're talking about connecting our workflows. And this is both physically and digitally. So physically, this looks like taking away that machine tending. So that will be using a platform to switch between instruments, to actually physically move the plate, so you don't have that manual interaction. And then it also means virtually. So that means having something that orchestrates all of this equipment. And then what this also allows is you to scale more effectively. And this is scaling without having to have more people because it is all contained in one system. So this allows you to schedule plates or samples through your system. And then by scheduling physically and digitally, you can actually increase your throughput. And some of the things I'm going to talk about kind of illustrate this quite well. But it also means that you should get better data out of it because you're going to be more consistent. Unfortunately, the less you get humans involved, the more consistent things become. We are naturally not consistent beings. So I guess the next question I would probably ask if I was watching this is like, do we need this? Is it necessary? And do people want this? So we did a conjoint analysis where we asked people these kind of questions, and this is just like a small subset of some of the things that they said. But 87% of people wanted to reduce the amount of hands-on time they were having to do, and especially for these more routine assays. 60%, when they thought about automation, they thought that they wouldn't actually have the space in their lab to implement it. And then 83% wanted to increase their sample throughput. And I've kind of categorized these into three little handles, I guess. So by increasing your throughput, you can unlock your lab's potential. By being able to implement this automation in normal laboratory space, you can unlock that space. And then by taking people away from doing things like machine tending, you can actually unlock your people as well. So I'm just going to introduce like the Link platform a little bit. There may be lots of people here that know about it, but just to provide context. So the Link platform is made up of, this is kind of our base unit. This is the core of what we offer. Obviously, this isn't all we offer, but this is the main components. So we call it the Automated Lab Bench. So it's made up of 3 components. So the robot and the rail. So this is what I'll be referring to as the robot arm. So what this allows is this robot arm on the end of it has a gripper that is able to handle SBS format consumables, labware. And what that means is that this robot is able to handle a lot of your samples and consumables. So it is able to move horizontally and vertically to cover a large space which it can access. And then with that, we have the bench surface, which we're calling the integration space. So it is literally what it says. It's just the bench surface. So, but this is such an important bit because this is where you will put all of your instrumentation. So all of the things that are actually going to enable the physical parts of your workflow. And then we have the transport layer. So the transport layer is also AKA the labware highway. So this is an area below the integration space where we have these things that I've detailed here. They're little shuttles, and these shuttles are able to hold SBS format samples, reagents, consumables. And what this layer allows is it allows movement across benches and long benches. And what this means is that you're able to utilize a different space for movement of your reagents, so you don't have to just rely on the robot arms. This becomes even more important when you start playing with the configuration of these benches. So if they're back to back, the robot it could hand over in the middle of the bench, but what this would allow is that you could then take from the front of the bench all the way to the back of the bench through to the front of another bench. So it just allows more flexibility. You can stage reagents. And it's cool. It's all based on magnets underneath this layer. And then these shuttles can hold reagents. They've got lights on them. They've got RFID tags on to just give you a little bit more visibility about where your samples and consumables are. So I'm just going to go into each one of those handles I spoke about and like actually what I mean by that and how the Links platform actually allows that. So unlocking your people might look like increasing your walkaway time. So obviously this robot is pretty much able to, in many ways, not all, you can't have a good conversation with it, but it's able to replace the human aspect, so the machine tending. So in another world, you might have to seal a plate, move it to a centrifuge, move it back to a liquid handler, or move it between a liquid handler and a plate reader. Well, the robot arm would allow that to happen automatically without any human interaction. So that means you've got that walkaway time, and it's not an inconsistent amount of time. It's going to be the length of your workflow. So that means you can go away for a whole day and do something else, because there are lots of tasks out there that aren't automated and aren't going to be automated and don't need to be automated. And then that's when we need the scientist to be available. Another big one, which again is quite personal to me, is actually reducing the manual handling errors. So things like liquid handlers do a really good job of getting rid of like pipetting errors or minimizing pipetting errors, you know, not putting the right volume on your pipette. Things like that don't happen with liquid handlers. But there's still parts of workflows where you might, as a human, have to pick up a plate, keep it in the same position and move it into an instrument. And actually, things can go wrong. You can have someone talk to you and you put it down and you get distracted and then you pick it up in the wrong orientation and then you put it into an analytical device, I guess, and then you get the wrong readout and you might not know you've done that. So I think it's a really nice way to never have to be in that situation where you feel terrible because you've done something silly like flip a plate, but it's so easy to do. And then another cool way that it can unlock your people is actually integrating all of the machine's initialization and like reading. So what I mean by that is, yes, it will move a plate to a plate reader, but then it will also, our software will then call that plate reader and say, read this plate, which I think is really cool, because in that picture I showed before, you saw there was lots of instrumentation, a lot of it would have been automation, but there were still people standing there, and that's because you still would need that people there pressing a button to say read. And same with liquid handlers, right? Once you've set up the deck, you still need to tell it to start. So that can all happen within the Link platform. And then unlocking your space. So the benches can be arranged any way you want, really. They can be back to back. They can go in an L shape. They can go in an L shape the other way. So they can just be arranged to fit your space. And another thing, if anyone's seen any of them at any trade shows or conferences, they are actually just the same size as normal laboratory benches. So if you have a space where you have 4 laboratory benches, then they can be replaced one-to-one with our benches. So when we say they fit a lab, yes, they fit a lab because they can fit the shape of a lab, but then also they can actually physically, dimension-wise, fit in the space that you have. And then also we can use them, a lot of the time we can use a lot of instrumentation that you already have. So you're saving space because you don't need to get additional instrumentation. So that's a really cool way to use your current laboratory space, by using them instruments you already have and you already like. And then also, you might see on this picture on the very end, there's a mock-up of a instrument on the layer where the transport layer is. So we actually offer a bench that doesn't have the transport layer as well, because that can allow you to really use vertical space. So you might not need to have reagents go all the way to the end of the bench because it goes up to here. So this means that you can put in another instrument there and just save that little bit more space. And again, one of our examples shows how useful this can be. And then unlock your potential. So I guess the way I take that is just being able to do more, just being able to do more science, right? And I think some of the ways that our platform allows that is everything's vendor agnostic. So we don't say, oh, we'd rather you use this vendor. We will do what we can to work with every vendor. The instrument choice should be yours, not ours. And then instruments can be swapped in and out. I think this is a really important one. So in big systems like this, sometimes you might need to reconfigure the whole system if you want to change one thing. With ours, you don't need to change everything, just to change one instrument. So it is literally taking it off, reconfiguring your workflow, but it shouldn't require reconfiguration of everything. And then same with scaling. So again, this will be in the examples, but you can start with a small system and you can add to it, and you don't have to reconfigure your first system. As part of our development process, we will find out if you want to future-proof anything, and we can leave gaps for things. But at the same time, if you want to start small, you can add to it. And the cool thing about that is it means that you don't need to commit to the bigger system before you're ready for it. So you don't need to wait until you're at full capacity before you can start introducing this platform. And then I just want to touch a little bit on the software as well. So we work on a node-based visual editor. So for each of these nodes, each of these nodes is an instrument. So it's quite cool. It's kind of like having a whiteboard and you drag and drop your instruments. And then within each node, you can set the parameters. So it's very visual, and you can join the nodes to represent your workflow. So you can set the parameters. So you can see here, this is a seal. The action is to seal, right? It's quite a simple one, but then if you go on to one of the other ones, there's different parameters such as the adhere time, so how long it's going to push down to seal the plate. So if there's parameters that can be set, you can set them within our software. Another really cool thing that I actually really like about this, that unfortunately I haven't shown here, is that you are able to create these things called subflows. So just say you have a step where you consistently seal centrifuge peel. You can create that into its own little subflow, set all the parameters, and then just slip that in, and it will know to call that each time. And then the other thing to note as well here is that for all of them robot movements, you don't have to program them in. The software will take care of that for you. So you don't have to say, to get to the sealer, to the vortex, I'm going to need robot 1 to do this. The software will work that out for you. And then the next thing I just wanted to show is kind of how this looks and how you might plan a batch. So again, this is for a similar, if not the same, for the same workflow. So you're able to input your batches here, and the software will work out the best way of doing that based on the parameters that you've given. So it can give you a duration. It will show you this kind of like timeline to see where you are. And then it will go, okay, so you need to do 2 plates and you need to prepare your qubit assay on this liquid handler. So once it's finished on this liquid handler, I know that it's free, and then I know that I can slot it in here. If you're interested more to hear about our software, definitely get in touch. This isn't like the focus of this webinar, but there's some really cool features and a lot to talk about. So my first example I want to talk through is our quality control workcell. And I guess the first thing I want to start on is like, why did we think it was a good idea to automate this? Why do customers think it's a good idea to automate this? And at the top here, I've put occupies day 3 of an NGS workflow. So I've said day 3, it depends on the workflow, right? It might be day 2, it might be day 4 if it's a long workflow. But personally, my experience is that when you're working on an NGS assay, there's always that extra day where pretty much the only job is to quantify, pull, and load the sequencer. But it's super important, but no one really wants to do it. It's very repetitive. You flip a coin, you make a rotor, because it's important but boring. So I think, and because it's so routine, it's low-hanging fruit for automation. A lot of it will be automated already, but it still has that machine tending where you have to move things from between instruments. So yeah, so that's why, like, I think from a workflow point of view, it makes sense. It's also really important for NGS assays because you need to know if your library prep's been successful, to know whether you go ahead and sequence it, which is an expensive thing to do despite current technology advancements. And then also you want to get the best utilization of your sequencing reads. If you could then go and put an unbalanced pool on your sequencer, then that can also be a lot of lost money. And a lot of lost time. Like, I've had to repeat experiments because of bad balancing of sequencing reads before. And it is a pain, especially when you get to the end of the process. It's taken you nearly a week to do something, and then you find out it was because you didn't quantify well enough. So this process is usually, the throughput is usually limited by not wanting to work outside of normal laboratory hours. It's set by the lab themselves, but it's not always something that you want to create a culture in your labs, especially in some discovery labs where people work too much anyway. And then also, it's just, if you're doing this manually, like, how much can a human do? How much can a human happily do in a day and do well? So I'm just going to talk through a customer problem and how we addressed it. So this customer were doing their library prep completely manually. And they were dedicating a lot of time to it. And they had increased their sequencing capacity. So they had a lot more to, they decreased their library prep capacity, so they had a lot more to sequence. So they needed to increase it. So obviously, like I said before, staff were getting full days to manual QC. And then they were finding the limitation was actually the amount of available staff, because they still had other stuff to do. And then as they were increasing that throughput through staff, they were also seeing an increase in error, which for all them reasons I mentioned is not ideal. So we were able to take, using our solution, we were able to take their 2 plates a day to 10 plates a day. And then this equates to 500K samples per year to 2.5 million. And then actually the full-time employee hours per week were 30. And then they ended up being down to half an hour. So how did we do it? So this is the QC system. So I'll take through kind of the labware journey or the sample journey. So on this system, we have, you'll see the labels there at the top, but we have a liquid handler, plate sealer, plate peeler, centrifuge, plate reader. All of these can be controlled by the Automata software, and they are all available to use for automation. So the user would load their samples in these plate stackers here and some consumables. And then they would go on to the liquid handler for the quantification preparation. So if it's for fluorescence, it might be setting up the dye, setting up the standard, setting up the dilutions. So that will happen on the liquid handler, as you'd expect. But for parts where they might want to do some vortexing on deck, but they aren't comfortable with having on deck vortexing without sealing, they might, and then you're able to, the robot arm is then able to move it to the plate sealer for sealing, move back onto the robot, and then move off to get the seal taken off, or moved off to get centrifuged and the seal taken off. Like, this is all available to them. And this is what they ended up doing. So all of the normal machine tending points of automated sealing, peeling, and centrifugation are taken care of. The only limit to the throughput of this system is actually how much you put here. So how much of the consumable samples you load initially, because it will run for as long as you allow it to. So on that software, you could put in 5 batches. You could make sure you have 5 plates worth of samples here, and then it will continuously run until it's hit them 5 batches. So your throughput is purely determined by the consumables loaded, and also how long you want it to run for. So it's a full walk-away solution. It's just adding at the beginning, and then this would be available for a user to come and take the completed samples out, to maybe if they want to keep the quantification plates for any reason, they could be kept in there. So what are the actual benefits of this to the workflow? Because, I mean, apart from it being, it's nice to not have to do things, there are other aspects that actually make it more worth time. And I've just included a GIF here of that solution running, to kind of give an idea of how these 2 robot arms can work at the same time as well, because that allows you to increase your throughput, because you could have 2 plates going through here. It's kind of like having 2 operators working on it. So things like time sensitive steps can be controlled. So adding a dye, as soon as you add that dye to your sample, you might want to move it to the plate reader as quick as possible, so it can incubate in there rather than out in the open. If you're not in the lab at the time and you're doing this manually, or you're doing it on a liquid handler and you're not there to move it over, you can get some inconsistency of how much light it's exposed to. But here, at least, as soon as it's ready, we're going to get involved, we're going to move that plate and put it into the dark plate reader. And then that thing I was talking about, how you can vary the batch numbers. So a system like this might feel like it's only worth it if it's running at full capacity all the time. But actually, if you have a day where you only have one plate to QC, then you only need to do one plate to QC. It can be varied by, you know, on the software and the consumables that you load. And then also that there's that bit about human error as well. You know, you're never going to misload a plate. You're never going to flip the plate before you read it and then find out once you've got your sequencing results back and everything looks very unbalanced. Again, that's a personal story. So that's our QC workcell. And then the other one I want to talk a little bit about is the nucleic acid extraction. So this is a beefy one. So quickly on why you might want to automate this. So obviously getting the nucleic acid out of the sample is an important first step for a lot of genomics workflows. And it's really important that you do it well as well. If you do a high quality extraction, you're more likely to get high quality data out. And you never want to be in that position where you're getting bad data out and you can't confidently say whether it was bad extraction or it's actually something to do with biology. And then also, if you have a high throughput extraction, that means that is now not the limiting step. So it allows you to be more high throughput throughout the process. And a lot of customers come to us saying, like, this is a bottleneck for us right now. And this is an opportunity to directly impact on that bottleneck. And again, so the throughput is usually limited by people, manual equipment, if they're choosing to do it manually. And then even with automation, you might be limited by just one 96 well plate at a time. So coming back to that customer problem. So their problems were slightly different to the previous customer. So they needed to increase their throughput to match their library prep throughput. They nailed the library prep. They were able to get plenty of plates through, get it onto a high throughput sequencer, but actually outsourcing all of their extraction wasn't practical, but they weren't able to scale up in the space that they had. So they needed that throughput increase, but they only had, I think it was 3 benches against a wall in the corner of their lab. And they'd already also spent a lot of time validating their chemistry as well. So that's not something that they wanted to change. So we worked with them to get their 4 plates to 16, 90K to 390K. And then actually they were able to free 2 of their employees up to do other things rather than extractions. So there was a few iterations of the solution, which I think is which makes it a really cool way to show the evolution of our system. So on this system, we have a large plate hotel. So this is capable of holding a few hundred consumables, depending on their size. And then we have this dispenser, so it's contactless. It's able to dispense to a plate, depending on the volumes, like in under 30 seconds. And then we have 4 bead, magnetic bead-based extractors down here. So what it might look like is you might take a sample out of here. You have your lysis buffer put on the dispenser, and then it will be moved down to one of the extractors. And then the role of the robot arm in this and the dispenser in the hotel combined is just to kind of feed the different buffers that are required for the extraction process to each 4 of these extractors. So, I mean, in this example, they use magnetic bead extraction. You could imagine another world, these could be vacuum, like vacuum or positive pressure modules. In this case, it was bead because that's what they were using and it worked well for them. So they were able to, so this robot arm is able to feed these consistently. And then you were able to get, if you were using a 96 well plate, and then you were able to get 384 out of this. So the software would be able to schedule these. They would start staggered, but they would all end within 10-20 minutes so of each other. So that gives you a 4 times throughput. Something you might notice about this as well is it is a very small system, and part of the reason we were able to do this is because we don't actually have a transport layer in here. It's a small system. You don't really need the transport layer. The robot arm has it covered. So we're able to utilize that space to put in more extractors, and then obviously that increases the throughput. And another thing I quite like about this as well is it offered a little bit of extra flexibility. So you could actually put different chemistries on here. They don't all need to be running the same thing. So you could have one that was extracting from blood. You could have one that's extracting from tissue or saliva or anything really, anything that is capable with these extractors. So you could have, you know, the same patients, but 4 different tissue types or something like that. Something cool like that, where it can all be combined at the end. So let's have a look. Oh yeah. So the upgrade to this, after speaking with the customer, is that with all of your extractions, you also need to QC them most of the time. Obviously, if you put badly extracted material into a library prep, then you're going to get bad stuff out. Or if it fails, you don't want your non-extracted sample to go all the way through library prep, because that's an expensive mistake to make. Oh, sorry. So we talked to them about trying to make a high throughput QC module to also complement the high throughput extraction, right? So the other thing to consider here as well was they wanted this extra functionality, but they still had limited space. So we had to come up with a way to be able to do this with just that third bit of lab bench space. And luckily, because we weren't using the transport layer, we were able to do that quite effectively. So on here, the extraction would all happen the same. It would happen on these 4 systems. And then when they were completed, our system would be able to move them onto this liquid handler up here. And then on that liquid handler, it would be able to create the dilutions for a qPCR, and it would be able to mix with the master mix. And then they would all be able to be combined and put into a 384 plate ready to be qPCR'd. So big advantage to this is you're getting a lot of data and a lot of extraction all in one go. And the cool thing about, and the cool thing about this is that these extractors down here, they could be 96, or they do have options to be 24 heads. So even if you wanted to do a larger volume extraction that you could only do with a 24 head, you could still do that. And then you could combine to a 96 well plate for qPCR. And then you could just do triplicates on your qPCR. So it can still be flexible, despite it having all of the devices on there. There's still flexibility within the devices. So we offer, you know, the integration of it all, but we don't take away any of the flexibility that these devices offer naturally as part of their service to you as well. So this was a really cool way of adding that QC, adding that functionality without increasing the footprint too much, which was a requirement for our customer. And also I just think it's quite cool as well. And then the next slide is just a way of kind of showing how dense this solution is. And I think this shows it quite well. So it's just cycling through like the different components, but it really uses every square inch of this bench to give functionality. And all of these benches on their own have a singular functionality in them. So you could even see this middle bench as just being a really dense way of doing extraction. It has all of the components there to do it. And then this second bench that appears, this adds full QC functionality, so ability to run them qPCRs. And then the one on the end is just highlighting the expansion of the system. So how the throughput can be increased and the runtime can be increased as well. That's why we've put such a big plate hotel on here, because the more reagents you put in, the longer the runtime. So we were working with someone who was already using benchtop extraction methods. And part of the things we like to do when we work with customers like this is just make sure, like, is this system hitting the goals that you want it to? And by comparing how they would have scaled up without our system. So to hit the 4 times throughput that we were able to achieve without our system, they would have had to have 4 full-time employees using benchtop extractors. And then they would have had to have 4 benchtop extractors. They would have had to have got more qPCR machines, so have at least 2 in total. And then all of this would come to about 350 square foot of laboratory space. And then with ours, obviously able to have 1 employee, because you only need that 1 employee to load the system and collect things at the end. 1 work cell, like the one I just shown you, and then that would be about 100 square foot. So you don't need to take as much space to get that full functionality. And then the next thing I just wanted to show you was just like an alternative solution we came up with. So obviously people might want to do different types of QC for their extractions. qPCR obviously takes quite a long time. It works well if you wanted to maybe use all of them samples at once downstream. Doesn't matter if qPCR takes a long time, but if you're okay with your plates coming out staggered, using a plate reader for fluorescence or something might be more preferable. So what this would allow would be very similar sample journey. You would put your lysis buffer on the dispenser, it would get moved down to extractor, and then it would move up to the liquid handler to prepare for the plate read, but then it would do the plate read 96 at a time. And then that means you're getting results in 5 minutes once your extraction is completed. So you'll just be getting a high turnover of results rather than a high volume, if that makes sense. So it just means that if it's more important to a customer to have your samples, your results sooner, ready to process your samples, because it might not be that they're all going to go on a library prep together. It might be that they need to be using some other kind of assay. Then this shows kind of a way of configuring the work cell differently, but actually ultimately it's delivering the same functionality. And then you also don't need as many extractors on there because things are being read once at a time. So you don't need to wait for 4 extractors, loads of plates to be together to make use of your qPCR runtime. So a lot of it is considering the schedule, and that's how we really hit the throughput. So I just want to finish talking a little bit about like how we get to this place with our customers. So I guess a lot of the things that I've been speaking about when I was speaking about our examples was kind of understanding their needs. So the first thing that we do when we talk to customers that are interested in using our platform is trying to understand what their throughput is. So, and I guess what the throughput is will depend on how we design a system. So like you saw before, I had that qPCR system, and then we looked at the plate reader system. They have same functionalities, but the throughputs would have been slightly different and for a different reason. So the main thing that we look at is throughput, manual interactions. So if they are literally like, I don't want to have to touch my system more than once a day, then that's important for us to know. And then also like how many staff they have, how much interaction do the staff want to have with the system, and all of them kind of things can be tailored to each solution. And then what we usually do is we take SOPs, we talk to the customer about them, ask questions, and then we'll come up with a 3D render similar to the ones that I've shown. So you can kind of visualize it, think about how it will fit into your lab. We can actually do some cool AR stuff where you can actually use an iPad to see it in your lab. And then we'll understand your user requirements and specifications. So we'll make sure that you know what you want from the system, we can deliver it. And then it all ends with a technical proposal and quotation. So if this is something you want to ask more questions about that you haven't come up with now, or you want to talk more personally about your solution, there's loads of ways to contact us through the website. So I totally invite you to. So I'm going to take a look at the chat now and see what questions we have. And thank you for listening. It's been fun. So a question that's been asked is, how do you ensure all of the integrated automation or the work cells remain sterile? Remain sterile while the workflow is running, UV lights, etc. Yeah, so a lot of these solutions I've shown haven't actually got these got any containment on them, but we do offer containment solutions. So again, this is kind of up to the customer. So we do have solutions where we would put our extractors in dead air cabinets. And for things like cell culture, we can provide like proper big laminar flow or class 2, is it class 2, class 3 cabinets. So we can offer that as part of the solution. So yeah, that's completely up to the customer how they do it. A lot of the actual liquid handling will be happening on liquid handlers for a lot of the steps. So you would be able to utilize the things that are already on the liquid handler. So if the liquid handler does have a UV function, you would use that. And then we would, you know, it's up to the customer to decide on the appropriate cleaning protocols. All of our benches are being tested for, have been tested for all common like lab detergents and stuff, and they're safe. But yeah, we offer that containment solution, which may involve using UV lights or whatever. Again, it's just it's a collaboration between what they need, what we can provide. But we do have some systems that have large containment enclosures on them to help with things like contamination. And then we have another question. Don't you have to watch the workflow for errors, etc. Though anyway? So our system of software does have error alerts on it and error handling. So if something does happen, you can get a text message, you can get an email, or it will come up on the actual the GUI, so like the graphic interface for the software. So you don't need to watch it. You can watch it if you want, but yeah, you will get the notifications if something happens. And if you can set it up so you will have control over you have control over what happens on them errors. And also if you need to have like a breakpoint and a manual interaction, that can also be programmed into the script. And then who has Link up and running so far? So we released a little while ago, we had a press release with the Francis Crick Institute in London. They've got an automata system, and it's running 3 workflows. So, and it's a genomics one, which is good. So it's running a CRISPR screening protocol. It's also running FFPE extraction protocol as well, and then it's also running a COVID surveillance protocol. So the Francis Crick, I think it'll be on our website. They've done a video with us explaining the system. So that's a really cool partnership that we've got going at the moment, and someone where you can actually see it running. And then someone's asked, if an issue occurs, how may I contact the assistance service? Yes, so we'll have, so when we have a system with someone, they will have a contact. It's a very collaborative process. So we have deployment engineers, we have customer success people, and whenever we deploy a system, you will know who you need to contact. So you will have a contact that you can talk through the issues, and then we'll decide what the next steps are, if we can help you remotely or we'll send someone out. So yeah, it just works like any other laboratory device, really. You'll have a contact, but we are quite attentive because we understand the complexities of the whole system. How will calibration be done? Will it be a routine? Yeah, so that's another question. Yeah, so calibration of system and robot training will happen obviously when it's set up, and then we will have a service contract with you depending on the needs of the system. So it might be different depending on what the application is. So this would be something that you would set up with your service engineer as part of your contract. So a lot of the calibration on our part is actually the robot training. So the robot knowing where the devices are, and the robot knowing where the devices are, knowing how high to go, all of that kind of things is something that we handle, and it will be routine. And then the actual calibration of the instruments is something that will be handled, we'll manage it, but we might have the assistance of like an engineer specific to that instrument who will come and do the calibration for that instrument. So again, it's all very dependent on your instruments and on your workflows. But and then I've got time for a few more questions. Is this easy to change if you have shared workspaces using it? Yeah, so I think if you have this in a shared workspace, the cool thing about it is you kind of saw how easy it was to make the workflows. So what you could do is you could actually, you know, as long as you have the appropriate instruments on the workcell, you can set up several workflows on the workcell. So you can set up several protocols. So if someone's not using it, and you want to run your qPCR on, but they would just run an extraction on it, it doesn't matter because you've already got the workflow set up. So you just have to put in the different consumables and press go. So you can set up several different workflows on the same workcell. It's not one workflow to one workcell, which gives you a little bit more flexibility, and it means it is a bit more viable to have it in a shared lab space, for instance. And then, oh yeah, and another question about vertical space. So can you also go up as well as going down? And yes, we can. So our only limitation is as high as the robot can go. Yep. Yeah, so it's as high as the robot can go. So if your robot can go up here, then you can have devices up to there. So what we have done in the past is we have stacked instruments that can be stacked, or we can place instruments on shelves. So we can just customly make some shelves for certain instruments. So yeah, you can go up as well as go down. There's no limitation apart from how far the robot can get up, which is quite, I think, I'm trying to think how high it is compared to me, but yeah, it's way above what I can reach, which doesn't really say a lot. Okay, great. Cool, I think that's all of the questions. Well, thank you everyone for joining. Like I said, if you want to follow up with any questions that haven't been answered, or you want to talk more personally about something, just get in touch. We always get back pretty quickly. Yeah, so thanks so much for joining. Thank you.